Thank you very much. I would like to say happy birthday PDB first. Using David Goodsell's iconic image, which obviously was presenting the molecule of the month almost ever since the PDB was created. This is those are amazing pieces of work, and if you want to know how they are generated, I advise you to follow David on Twitter, because every item that he makes, he documents the entire process of it. So it's really quite mesmerizing. Now, David has been extremely influential for my own thinking about structural biology, particularly for one reason, which probably he never intended in his images. But what he managed to convey, at least to me, is the amazing complexity in which the structures and the molecules that we're all interested in which they function. And that is, of course, the intracellular space. And while this is an artistic representation, I think we're getting ever closer with experimental methods as well as computational methods to come up with a true representation of what the cytoplasm of a eukaryotic or a prokaryotic cell really looks like. And we're beginning to understand that this amazing fidelity that biology has functions in an incredibly complex environment. And I just want to remind you that every protein that we study, every protein that we look at, has evolved to actually function best in this kind of environments over millions of years of evolution. So the fact that proteins act in cells is not just an unfortunate coincidence that makes our life difficult in order to study and analyze them, but this is really where they work best. So as soon as we take them out and we're trying to manipulate them in pure buffer or try to force them to grow into a crystal, we're actually doing something to a protein which really it doesn't want to do, okay? So what unites all of us here in this particular audience is that we are trying to understand biology at the highest level of resolution. And we may have taken up different types of disciplines over our careers to study structural aspects or dynamic aspects of proteins, but really we do want to understand biology. And over the recent years, what we all have witnessed and seen is that we will be able to not only understand biology in vitro and purified components and systems, but that we will eventually be able to study biology actually where it happens. And this is to a large extent inside cells. Now, I want to take you back to 2016, when I had a couple of extremely interesting discussions with Carol Robinson. And at that time, Carol was establishing a new department at Oxford University, and we were discussing her vision that was to put together physical methods that had previously worked on purified and isolated samples, and to combine those methods because many of them had reached a level of sophistication to also perform those types of analysis directly inside the cell. And what was most remarkable for me in those discussions was that Carol actually used the analogy of when the Laboratory of Molecular Biology, the LMB, was founded in 1962 by the Medical Research Council. And because it was founded basically on the promise that the first crystal structures will define a whole new research area that we now understand obviously has become structural biology. And what she said is you have to think back at that time, because the first crystal structures were nothing like what we nowadays think about, typical structural biology. And to have the vision, the imagination, that what started as an effort that looked like this and really become what is structural biology today was really remarkable in terms of insights and foreseeing that the Medical Research Council signed up for in times where, in order to get to those rather rudimentary structures, you needed amazingly basic, but at that time truly advanced methods of computational resources. So the code for those types of ancient computers were actually printed on paper you know, the only one who probably understood what this machine was doing was Max Berutz himself, you know, feeding those paper strips into the computer in order to do simple Fourier transforms, you know. And this is how structural biology started. This is how extracrystallography started. And look at us today and how far we have gone with this. And so Carroll said that, you know what, now is the time when many of those physical methods can really be, you know, used in a cellular context. And what she wanted to do is she wanted to essentially establish the equivalent of something like the LMB, but she wanted to call it, you know, a true laboratory of cell biology, so something like the LCB. And the reason why I tell you that this was all pre-Brexit, and this was actually pre-Brexit referendum, is because this vision that Carroll had sort of obviously, you know, got a little sidetracked or, you know, modified, let's say, by the outcome of Brexit. What still happened nowadays is that there is a new department at the University of Oxford, and this is now the Kavli Institute for Nanoscience and Discovery. And if you go to the webpage of this new department, you will see that at least some of those ideas truly materialized in this new department. However, what is, in my opinion, probably even more important is that the Wellcome Trust has funded a PhD program, and I think this is the first PhD program of this kind, which is truly a cellular structural biology PhD program, where you can experience those different methods and really learn how to integrate them and how to come up in a synergistic, complementary type of fashion about, you know, the inner life of the cell, so to speak, in his protein and biomolecular components. So here's a little exercise that, you know, I was playing. I invite everyone for you to play the same. It's like if you were tasked to sort of come up with a blueprint for the structural biology of the future, which would be the methods that you would like to ideally see combined in one place in order to create the synergies to sort of really write or be, you know, in front of the new wave of structural biology and sort of to shape the future rather than to watch it happen. Okay? And so here's my take on this. You may, you know, get some ideas out of it. You may agree, you may not agree. This is all perfectly fine. But let's play this game together. So what I think, at the top of the table, at the head of the table, clearly for me, cryo-electron tomography, cryo-STEM, electron microscopy-based methods that work in a cellular context. So we've seen remarkable breakthroughs over the last years, spearheaded almost entirely by Wolfgang in Martinsried, Germany. So here is really a pedigree, you know, it all started in Europe, okay? I think we should be fully aware of this, and we should capitalize on the fact that the world expertise for this type of technology is exactly here, right with us. Now, intuitively, you will immediately pair those type of methods with high-resolution imaging type of approaches in optical microscopy. We have amazing new developments in microscopy technology, software for microscopy types of technology development. If you just think of MinFlux, for instance, what MinFlux is currently able to do, and I understand it's a difficult method and it's not easy to set up and to really start counting molecules in the cell, but you know, this is exactly the way we're going. So to combine in a correlative type of fashion those new technologies in optical imaging with cryo-EM, clearly, you know, this is a very strong axis in the future Department of Structural Biology, so to speak. I want to include, and this may come as a little bit of a surprise, I also want to strongly recommend to think about mass spectrometry and to really integrate the types of applications that Yuri has told us about, or native MS, in a future department of structural biology, because I think it will be those complementary methods that will embellish also those imaging type of techniques with the kind of information that we probably might not be able to see a priori. But as a discovery tool, extending possible complexes inside the cells, and thereby creating new masks, for instance, for subtomogram averaging, including with AlphaFold, for instance, just predicting structures. You know, this is really of immense potential, in my opinion. And so I think, in my book, definitely mass spectrometry has, you know, a clear chapter to write in this endeavor. Now here are a couple of spectroscopic techniques that you may not be too familiar with. I mean, this is the kind of area that I'm coming from. But there have been also almost a decade or more of fundamental developments, both in solution NMR inside cell as well as in solid state NMR inside cells. For solid state NMR, Utrecht, for instance, is one of the epicenters of those types of developments. And the added benefit for those spectroscopy methods are actually twofold. Let me talk about solution NMR first. Solution NMR truly works in solution. Now the inside of the cell, in essence, is a liquid. This is really the kind of environment where solution NMR shines. We don't freeze cells, we really work in the cytoplasm. We work at ambient temperatures. In other words, we work at exactly the temperature where the cell performs its function. This is a non-destructive method compared to many of the others. In other words, we don't shoot high energy electron beams on our samples, but we actually pulse radio frequency pulses, which are completely benign. What that means is that while we study our samples inside cells, we don't destroy them. We can follow them over extended periods of time. And as you will see, we've built small bioreactors inside the NMR tube to even control the metabolic conditions that those cells have. In other words, we can keep them perfectly happy and healthy. We can feed them, we can challenge them, we can synchronize them, we can trigger cell cycle release, we can do lots of stuff while we keep recording atomic resolution NMR spectra. And what we will be able to do in the end is we will be able to assemble time traces of structural events that happen inside cells. And this is unique. Being able to add a time axis to those kinds of analysis is something that NMR can provide you with simply because of the fact that we don't destroy our sample. Now, solution NMR works really well on dynamic parts of a protein or entire proteins which are highly dynamic. Now, Ben referred to them as intrinsically disordered protein. A biologist would probably prefer to call them, regulatory sequence elements and otherwise folded proteins. And those regulatory sequence elements are essentially the functional keys of proteins. This is where post-translational modifications happen. This is where you can really, turn an activator into a repressor or a cytoplasmic protein into a nuclear protein simply by establishing new chemical groups on individual residues, and these kinds of codes can reprogram the entire function of the protein. Now, if you have an atomic resolution method like NMR, you can actually see those things happening inside the cell. You can follow them in real time, how they're being established, what they lead to structurally, how they're being removed again. So all of those kinds of things are possible with NMR. Extend this to solid state NMR, and you can even do it on proteins that are embedded in the membrane. And again, I urge you to look at the work by Mark Baldus and Markus Weingart at the Byford Center here in Utrecht. Those guys have really pioneered the application of solid-state NMR for biological samples. So remarkable developments also in the spectroscopic techniques. Included should be also electron pyromagnetic resonance spectroscopy. Why? Because, remember when Ben told you about the molecular ruler that Fred does provide, this is exactly the same that also EPR provides. So using spin pairs, you can actually measure long-distance distributions in folded as well as intrinsically disordered proteins. So it's really a ruler that goes over length scales that are much larger than what NMR can provide. So it's truly complementary. It can work in solution as well as in the solid state. You can freeze your cells or you can keep them at room temperature. EPR provides very important complementary information to NMR, both in liquid and in the solid state. And that brings me to the next one, because EPR is an ensemble method and a ruler, whereas single molecule is a single molecule method. And the same kind of ruler applies. However, now you start counting events on the single molecule level, and you can add up those events to get an ensemble statistical average of how a certain protein behaves inside the cell. The huge advantage about single molecule threat, however, is that you can do it in a localized fashion, which you cannot do by NMR and EPR. In other words, you can focus your confocal spot exactly where you think the action is going on, whether this is in the cytoplasm or whether this is in the nucleus. And you can actually measure distances and dynamics in different parts of the cell. Clearly, computational methods will play a major role in any sort of department. Whether you think of AlphaFold or whether you think of Relion, which we nowadays with Georges Connect as a cryo-EM tool, but it initially started as a software package that enabled new breakthroughs in cryo-EM. And this is of course also true for light microscopy imaging. There are huge amounts of development in software tools to manipulate images, reduce the noise, gather more information that is actually there in the data, but that we might not immediately have access to simply because information is too convoluted with noise. So any department of structural biology of the future will need a very strong computational unit. And within this unit, you can actually have several subunits. I think it's very clear, including AlphaFold, that this is where the future of structural biology will be going. So this ought to be included in my book of the blueprint for the structural biology department of the future. Finally, and this may also come as a surprise, I would strongly recommend to include scientific visualization. And not for the artistic sake of it, but to actually create a visual story of the types of an accurate, scientifically accurate story of the types of results that all of those methods combined provide. And again, I might remind you that David Goodsell, Gianni Di Sava, Sean O'Donoghue, those are all scientists that are linked to research universities and have their independent groups there in order to integrate and visualize the kind of data that those research units perform and to tell those scientific stories in an accurate sense. This is not about creating beautiful images. This is about communicating results which might probably have a time axis, and we need to tell, conformational rearrangements in a proper manner along the time axis. And even if, ND simulations give us a hint of where things are going, in order to tell the story properly, we need people that have a scientific background, but at the same time know the kind of tools of how to tell those stories properly. Now, when I moved to the Weizmann Institute of Science in 2018, I thought, this is actually a place where many of those, items in my list of a really good structural biology unit were naturally present. And so I know that many of you will recognize faces in your specific disciplines, and it is true that in principle the Weizmann Institute is like one place where you already have many of those experts working exactly in the same spot. So you might ask, how has it been? 3 years later, how productive have you guys been working within this amazing kind of environment? And to be honest, the answer is not too productively, because once we arrived, COVID hit, and then things went down sort of from there on. I do understand that, of course, everyone suffered through the pandemic, and that many places and many labs in Europe were closed down or restricted in access. Movement between countries were not possible. All I can tell you is, if you're in Israel and this country sort of closes itself off, that's it. We're unable to recruit. We were unable to. Well, we could interview via Zoom, but then we could never, get people over. The lesson that I learned in this, and I think this is also the kind of Brexit lesson that we learned, is like science lives by exchange of people and minds and talents. As soon as you isolate yourself, either because of a pandemic or because you feel that you have to leave, certain other groups of people, science is going to suffer. So we should avoid those types of consequences. So let me tell you a little bit just how does IncelNMR, IncelEPR work. It's actually surprisingly simple. Let me take you back into the cell. This is a truly crowded place. The reason why you guys do get some information out of this picture is because everything is colored, right? So individual components, you can identify them, you can easily separate them, but of course in nature nothing is colored, so it would look more like this. And this still has enough contrast for the people in the first row to actually see differences, whereas people in the last row, I bet you only see gray. That's exactly what I'm trying to accomplish here. because clearly by tomography, for instance, we would like to have the contrast of the first row, but in essence very often we have the contrast of the last row. And of course that makes it very difficult to identify individual structures in this soup of cytoplasmic protein. So what we need to accomplish is very simple. We need to specifically highlight those components in the cell that we want to study. And I know that you guys in the first row see what that is. You guys in the last row will probably only see some yellow dots, but this is enough for making the point that if those yellow dots are the protein that you're interested in, you will be able to selectively study them against the backdrop of everything else in the cell. Now that doesn't mean that the cell is empty. I just remind you that this is what we do in microscopy every day, right? We use fluorescent dyes, fluorescent labels to selectively visualize one component against another, against the backdrop of everything else in the cell. That doesn't mean that the cell is empty, like the black background here. It just means that you only visualize the component that you're interested in. So how can we specifically visualize by NMR one protein within the soup of all the other proteins that are there, for instance? The answer is very simple, by just making them slightly heavier. What I mean by that is we use stable isotope labeling to turn a protein which naturally occurring is not visible. We label them with stable isotope, a protein, and suddenly the NMR spectrometer sees it, because those are the types of samples that we use in classical NMR as well. We always need to isotope label whether we work on a pure sample and want to determine a structure in vitro, or whether we want to look at the single protein against the backdrop of everything else inside the cell. The key is to add isotope labels to the stuff that we're interested in and rely on nature not using any of those isotopes for everything else that is there inside the cell. So now the question is, okay, that sounds reasonable, but how are you going to specifically make sure that only your protein of interest is being isotope labeled in the cell and nothing else? That's easy too, because what we do, we actually produce the protein outside of the cell in bacteria that we grow on heavy isotope labeled media. We purify the protein, it's a perfect in vitro sample if you so wish, and then we bring it back into the cell. How do we do this? Well, there are many ways. What becomes sort of, at the moment, a little bit of the gold standard is to use electroporation, especially if you're after mammalian in-cell NMR samples. Now, electroporation, you may know for DNA, RNA, it actually works really well for proteins too. So here's the procedure. So here you may have a cell line that has very little of the protein that you're interested in, or you've actually down-regulated it by siRNA because you don't want to spoil the signal that you get for an isotope-labeled protein but with endogenous background of the same protein, and then you use electroporation to just deliver the protein of interest. And in this case, the protein of interest is isotope-labeled. You collect your cells, you either put them directly in the NMR tube, or nowadays, as I told you, we use miniaturized bioreactors where we constantly monitor the metabolic state of the cell slurry that now all contains your isotope-labeled protein of interest. And we adapt and feed and make sure that cells are happy and viable and doing well. And then, if you're lucky, you get in-cell NMR spectra. Again, the first row will see lots of red peaks. The last row will only see lots of red. Red is good because red is essentially NMR signals coming from a protein directly inside cells. And here are 5 different cell lines. The great advantage of electroporation is you can manipulate many different types of cells, standard cells, more sensitive cells, but delivery usually for every protein is fairly efficient. Now you're already beginning to see, I can tell you're scanning those individual panels for differences and similarities. You see every cross-peak here responds to an individual amino acid of this protein. You can follow how similar or how different each and every residue behaves in those 5 different cell lines, and you begin to see interesting features that are unique for cell lines or that are more common for different cell lines. But this is exactly what we are after, because every difference tells us something like about the biological story that may go on in one cell line and not in the other, or things that happen in all the cell lines. Now you can play interesting games because you control the conditions of your cells. You can expose them, for instance, to oxidative stress. And if your protein inside the neuron happens to be one of those typical amyloid proteins, you can essentially trigger aggregation. And you can do this also in the NMR tube. And then what you will see, you start from a lot of red signals to very little red signals, because what happens is you start from a highly dynamic arrangement of your amyloid protein, which is not yet aggregated, but then over time the protein begins to come together, and more and more solid parts are beginning to form, because maybe the amyloid core starts in a certain region and then zips up into other parts of the protein, and this is exactly the kind of information that you can read out with those time-resolved in-cell NMR experiments. And again, I think this is unique, because not only can we look at the final stage of those aggregate forming processes, but we can look at the entire process. And we can interrogate exactly what is the contributing role of each and every residue in your protein of interest. And if you were to want to design an inhibitor against this aggregation process, of course you would try to target sites in the protein which probably come together early and prevent the entire process altogether, rather than sites which are probably late, where already the cascade has sort of set off and is in full swing. Now, clearly there are challenges. And the most obvious challenge is that, for spectroscopic methods especially, it's actually really hard. It's not easy to create a high-quality in-cell NMR sample to make sure that the cells are happy and that the NMR signal quality is still of decent amount so that you can really create interpretable spectra. So I always advise people, if you want to give it a try, don't think that this is just going to be a quick experiment and you will have the full in-cell story, because it isn't. The advantage, though, is that you can use surrogates rather than a complete in-cell NMR sample. You can, for instance, work with cell lysates. Those are easy to prepare from different sources. Those are homogeneous cellular environment solutions. You can just add your isotope labeled protein, and you will again use the stable labeling as a filter to only see your protein even in the cell lysate. And very often the biological activities that you are after, they're actually manifested already in the cell lysate. So you might not even have to go to a complete intact in-cell NMR sample to get the kind of biological information that you need. So the true thing is quite challenging, whereas to create preliminary data that is rich in information is actually very, very simple. So I think it really balances out nicely. Here's a problem which I think is much more prevalent, and that is really the fight of physics between biology. And what I mean by that is that many people in spectroscopic techniques are trained in the discipline of physics, and they appreciate the powers of their method, especially knowing that ideally we would like to work with homogeneous samples in isotropic type of environment, because only then we can really harness to retrieve precise dynamical information, for instance. By definition, this is not what a cell is. So as soon as you go into the cell, you know that your results are going to be messy, no matter how accurately and precise you set up your experiments and analyze your data. Okay? So as a spectroscopist, trained physicist, at some point you have to let go of the pure world, and you have to acknowledge as well as embrace the fact that once you start measuring in the cell, things are not going to be as perfect as you usually would like them to have. And I can tell you that this is a real issue for many people in the community. So it's very hard to let go of the pureness of a true physical tool for the sake of being able to study fundamental biological processes. I'm not saying that this is any different in cryo-EM, but I think people are much more understanding that, of course, cryo-EM or cryo-ET works inside cells, whereas NMR, EPR, and also single molecule FRET very often doesn't. The other thing is that even members of your own community may look down on you, because they will tell you, what you're doing, is strictly speaking not accurate. You cannot determine relaxation rates inside a cell because simply the environment is not homogeneous. And of course, those people are correct, right? I understand the drawbacks, I understand the caveat, but what is the alternative? The alternative is not to do those experiments at all. So either we learn something fundamental, and it might be, having some issues, and not be as precise as a pure buffer sample, but at the same time, at least we're going somewhere. than not doing those experiments altogether. The other thing is the biologists. I think we need to understand that our new audience is really the biologists. And as spectroscopists, we need to make sure that the biologists take our results serious. In other words, we need to perform the controls that satisfies the biologists and not necessarily only the spectroscopists. We need to choose the cell lines that we're looking at which are relevant for the biologists, and not necessarily because those are very robust cells and we can easily do experiments on them. Okay? So we need to commit to embrace cell biology. We need to commit to establish new biological systems which might be a lot more challenging than HeLa cells. But HeLa cells are really messed up cells. And a lot of biologists will tell you, whatever you see in HeLa cells, it doesn't really mean much because, just count the number of chromosomes in HeLa cells and you will realize that this has nothing to do with the diploid chromosomal set of a mammalian cell anymore. So I think we need to also embrace that. Challenge us also in terms of biology, because we want to be taken serious with the biologists. Enough of those challenges. I want to thank the people that really take upon themselves those challenges. This is actually the team just before it moved to Weizmann. Again, it has been a ride, not only for us in Israel, but I think for everyone over the last 2 years. I hope to be beyond that COVID type of mess that we had all experiencing, and that we will be able to see each other more regularly and in person in the future. And with this, I thank you for your attention, and I'm happy to take questions.